The topic of today's lecture is genetics of bacteria. Genetics. First of all, we should remember next terms. First, genetics is the study of genes, their structure and function, heredity and variation. Second, genomics is the study and analysis of the nucleotide sequence of DNA. Genome is the complete set of genetic information for the cell. DNA is the storage of biological information, excluded RNA-consistent viruses, where the storage of biological information is RNA. Gene is a locus or region of DNA which is made up of nucleotides, structural units of the nucleic acid, and is the molecular unit of heredity. Genotype is the part of DNA sequence of genetic makeup of cell and therefore an organism or individual which determines a specific characteristics, phenotype of that cell or organism or individual. Phenotype is the composite of an organism observable characteristics or traits such as its morphology, development, biochemical or physiological properties, phenology, behavior and products of behavior. Storage of genetic information is DNA or RNA for some viruses. DNA is a molecule composed of two polynucleotide chains that coil around each other to form a double helix carrying genetic instructions for the development, function, growth, and reproduction of all known organisms and many viruses. RNA is a polymeric molecule essential in various biological roles in coding, decoding, regulation, and expression of genes. Each nucleotide is composed of one of four nitrogen-containing nucleobases, such as cytosine, guanine, adenine, and thymine for DNA, and uracil for RNA, and sugar called deoxyribose for DNA and ribose for RNA, and phosphate group. Transfer of genetic information is based on three processes. Replication is synthesis of new DNA. Next step, transcription, synthesis of new RNA from matrix DNA, and synthesis of protein is called translation. Why we use bacteria in genetics experiments? Because bacteria is comfortable model for genetic studies. Bacteria so quickly multiply. Cell division occurs every 20 minutes. High rates of spontaneous mutation, one mutation at 1 million cells. Several mechanisms of bacterial DNA transfer from one cell to another cell via phage, conjugation, and transformation. And presence of additional genetic elements such as plasmids, transposons, IS sequence, and others that can transmit the encoded their other properties of bacteria. The genetic material of bacteria concentrated in next structures: intrachromosomal, nucleoid, and extrachromosomal heredity factors such as plasmids, retroelements, integrons, transposons, mobile gene cassette, IS sequence, and phages. Nucleoid has circular double filamentous molecule of DNA, located in the cytoplasm, contain genetic information, and it is prototype of eukaryotic nucleus and absence of nuclear membrane. Plasmids are small and extrachromosomal DNA molecule within a cell that is physically separated from chromosomal DNA and can replicate independently. Properties of plasmid: has additional DNA structure, circular structure, has from 1 to 30 copies in one cell. Replication occurs independently of the chromosome. Transmissivity is the transfer properties from the bacteria to other bacteria via plasmids. Can eliminate the disappearing, mutate, recombine, integrate, joining with the chromosome to form an episome. Functions of plasmid: regulatory is compensation metabolic defects. Encoding is making information about the new features into the bacterium. Classification of plasmids. Fertility F plasmids, resistance plasmids, cold plasmids, degradative plasmids, and virulence plasmids. That these plasmids play important role for development the infection disease. Transpositional genetic elements consist in 5 groups: transposons, I sequences, integrons, and mobile gene cassettes, retroelements, moderate and defective phages. Transposons is a DNA sequence that can change its position with the genome, sometimes creating or reversing mutation and altering the cell's genome size. Make a duplication, deletion, and inversions. They can be located in an isolated or integrated state, but not capable of independent replication. The gene is characterized by the same genes as plasmid, determining some phenotypically expressed antibiotic-resistant toxin formation. The large molecule of DNA consists of a structural gene able to navigate the chromosome. The significance of transposons. The first, mechanism of enhancing genetic transfer, spread from one organism of species or to another, antibiotic resistance, and genes manipulators. As sequences is short for fragment of DNA, don't have a structural gene, consist genes that are responsible for transposition, the ability to move around the chromosome and integrate it into its various sections. As sequence functions, coordination or interaction of plasmid, transposons, temperate phage, and bacterial chromosomes. Regulation of the activity of the bacterial chromosome genes may inactive the turn-off of gene or exhibit the effect of promoter. Induction of mutations such as deletion or inversions and duplications when moving after insertion into the chromosome. Integrons and mobile gene cassettes. Integrons are genetic elements that contain determinants for the components of the site-specific recombination system, then uptakes and promotes the expression of mobile gene cassettes. The gene cassette is a discrete mobile genetic unit that is free covalent circular DNA molecule that contains the one or more than a recombination site, belonging to the 59 base element family of sites. Retroelements are mobile genetic elements which are capable of self-reproduction through RNA intermediates, which are transcribed via reverse transcription of DNA using the reverse transcriptase enzyme and integrated in the new location in the host genome by the integrase enzyme. Moderate and defective phage can account for up to 20% of the bacterial genome, incorporating in the bacterial chromosome in the form of prophage provided then cause lysogenization of bacteria which can acquire new properties. Next, the topic of our lecture is variability of organisms, are changes in the properties under the influence of various factors. Variability of the organism divided into groups: phenotypic, non-inherited, and genotypic, inherited. Phenotypic consisting of 3 properties: morphological properties, cultural properties, biochemical properties called dissociation, and genotypic inheritance, mutation, and genetic recombination, such as transduction, conjugation, and transformation. Phenotypic variation is the physical expression of various characters by a bacterial cell in a given environment. These properties are determined not only by its genome, but also its environment. Phenotypic variation, for example, synthesis of flagella, Salmonella typhi or pathogen of typhoid fever, is normally flagellated, but the flagella are not synthesized when grown in the phenol agar, and is reversed when subcultured from phenol agar into broth. Synthesis of enzyme. Another example Example of environmental influence is synthesis of Escherichia coli, the enzyme called beta-galactosidase, necessary for lactose fermentation. But this actual synthesis takes place only when it's grown in the medium containing lactose, such as McConkie or endomedia. Mutation is the permanent alteration of the nucleotide sequence of the genome of an organism, virus, or extrachromosomal DNA or other genetic elements. A mutation can be divided in three groups: by origin, spontaneous or induced; by localization, in chromosome or in gene. If localized in chromosome, we called inversion, deletion, duplication, insertion, and transposition and translocation. If localized in gene, we can differentiate point and frame shift. By effect on the variability of the bacteria, natural, conditional, lethal, and lethal mutations. Genetic recombination is transfer genes between bacteria. We need two cells, donor and recipient. Transformation is genetic alteration of a cell resulting from the direct uptake and incorporation of the exogenous genetic material, or exogenous DNA, from its surrounding through the cell membranes. Free DNA. Transduction is the process by which foreign DNA is introduced into the cell by virus or viral vector. Conjugation is the transfer of genetic material between bacterial cells by direct cell-to-cell contact or by bridge-like connection between two cells. Needs sex pili. Transformation. If you will see on the picture, we have donor, free DNA, and recipient, cell consisting chromosomal DNA. The free DNA and transfer into the recipient cell via membrane, and we can see the recombination between donor DNA and recipient DNA. Other part of the free DNA will degrade it unrecombinated, and we can see the integrated of donor DNA in the chromosome, this B group. As a result is genetically transformed cell. Transformation example by Griffith. Griffith studies the transformation via pneumococci strains. He inject virulent bacteria in mice, and mice died after 1 day. He inoculates the pass from the mice on the nutrient agar and detect the virulent colonies. Next group, when he injected avirulent bacteria in the mice alive. And when he inoculated the strains from the mice on the nutrient agar, he detected avirulent bacteria. When he prepared the heat-killed virulent bacteria and injected in the mice become healthy, but no colonies on the nutrient agar. When he prepared avirulent and heat-killed virulent bacteria and injected in the mouse dies, and on the nutrient agar he detected colonies of avirulent strains and virulent strains. Transduction. For the transduction we need recipient cell and bacteriophage as a donor. In the first stage, bacteriophage inject DNA. phage enzyme breaks down the host DNA and start the cell creates a new phages, including phage and host DNA. Next, transducing phage insert donor DNA and donor DNA included in recipient chromosome due the recombination. Conjugation. For this process, we need sex pili. Donor cell produce pili. Pili is attached to the recipient cell and brings the two cells together. The mobile plasmid is nicked and a single strand of DNA is then transferred to the recipient cell. Both cells synthesize a complementary strand to produce a double-stranded circular plasmid and also reproduce pili. Both cells are now valuable donor for the F factor. That's all for today. Thank you for your attention.